Hi, everyone, and welcome to today's Seed World Retail Strategy webinar. My name is Mark Sinkwitz, and I serve as editor for Seed World Canada. And today I'm happy to be your host. Our theme today is how scientists are tackling evolving pests and pathogens. We'll hear from three scientists who are going to talk about their work in oilseeds, cereals, and pulses, and how they're building this next line of defense against insect and disease threats. Before we begin, of course, I would like to thank our sponsor, CCAN, for their support. Our guest speakers today are Tyler Wist. He's an entomologist with Agriculture and Agri-Food Canada in Saskatoon. He works on making wheat resistant to the wheat midge. Sabina, hello, Sabina, is a pulse pathologist at the University of Saskatchewan Crop Development Centre, working to make pea and lentil resistant to root rot. And last but not least, Horatio Lopez Nicora is a plant pathologist at Ohio State University who works on soybean cyst nematode, which is always making headlines here in Canada. It's something that has been found down south and is slowly making its way north. So it's great to have you on, Horatio. Thank you very much for your time. Again, our webinar today is sponsored by CCAN. It is being recorded and will be made available on our website after the webinar. Also, if you have a question, feel free to type it into the YouTube chat box at any time. You don't have to wait until the end of the webinar. Feel free to ask your question at any time. We're going to start off today with Sabina, but Horatio and Sabina, thank you for joining me live. Tyler, his talk is pre-recorded, so I will be presenting that midway through our webinar. We'll talk with Sabina first, then we'll hear from Tyler, and then we will speak with Horatio. Now we're going to start with Sabine. So please hang on, Horatio. I'm just going to take you off screen here for now, and then we will see you again in a little while when we do your talk. So feel free just to hang out and relax, sir. Let's see. There we go. How are you today, Sabina?
I'm fine, thank you very much. Beginning of a new week, and it's a little bit cooler, so that is always quite nice. And as a plant pathologist, I have to admit I've been quite happy because we have had rain.
Well, and it's always nice when it's a bit cooler. What I know about the disease triangle is that warm weather definitely favors a lot of these diseases and pathogens, especially warm, humid, wet weather. And so we've got some cool weather. I suppose that's always a good thing. Now, I interviewed you recently. I wrote an article about you in regards to your work on root rot in pea and lentil. And this is something that's caused major yield declines and economic losses. Now, you're working on a project at the Crop Development Center that recently received over $4 million from what's called the Strategic Research Initiative. Before we talk about exactly what you're working on, how do you plan to utilize these funds to tackle root rot in pea and lentil crops? That's a lot of money. How is this going to be used?
Yes, it is a lot of money, and sort of the money obviously came with a certain framework already. So we were really asked to get together as a larger group of scientists to try to tackle this problem in a very comprehensive way. So really look at it from different angles. And maybe you can sort of show my slides because, okay, yeah. So does that allow me now to act?
Yeah, you should be able just to click on the next slide, and we'll see you switch. It should. I'm seeing your mouse move. So if you click on the next slide, it should change to the next one.
Yeah, let me. Sorry. Okay, so we were asked to look at it, as I said, in a very comprehensive way, and so I put together this slide here, which sort of shows the building blocks of this project. And if I start at the top there, with the orange building block, so obviously resistance breeding is a big part of it, and that is where I am mostly involved in. There's also a small component to look at developing tools for gene editing. We are not right there yet to be able to edit root rot resistance, but we do need the certain tools, and so some money will go to that. There is also quite a big component looking at exploring the pathogen biology, its diversity, its genetics and genomics, because that is really important to inform plant breeding and certainly gene editing, but then also disease development in the field. And then we also finally convinced all the participants from the funding side that it's really high time that we start initiate long-term crop rotation studies, because for one pathogen in particular, we know that crop rotation is really important, and we don't really know, okay, for how long do you have to sort of walk away from pea and lentil before you can recrop it, and so that will be initiated here. We will also look a little bit at biocontrol and other products that are on the market and see whether, if we combine them and use sort of the first generation of varieties with improved resistance, whether that will provide short-term solutions for farmers. And then last but not least, there's also one component where we will look at a new approach to sort of protecting crops, and that is RNA fungicides.
Yeah, like there's so much work like to be done in this area, and like you've got to investigate all these different things, and that's kind of what I wanted to ask you next was, what are the biggest challenges that are faced in helping develop resistant varieties? Because it's obviously you've got so many things you have to investigate and look at, and that tells me that yeah, there's a lot of unknowns, and that's obviously one of the big reasons why we need more varieties to have better resistance to root rot. So what are some of those big hurdles that you face?
Yes, so I think the biggest problem with the root rot is it is not just one organism we are dealing with. So it's a mix of several pathogens. Most people have probably heard of Aphanomyces eutychus. That is in many ways sort of maybe the most problematic one, because we know probably least about it, and there are really no effective chemistries against it. That's the one that can survive in the soil for really long periods, which is why there is this recommendation to have 6, 8, maybe 10-year rotations without pea and lentil. But then you can see here on this slide, there is also a whole catalog of different Fusarium species. And I mean, if you are primarily a cereal grower, you will probably recognize some of the names. So they can infect pulse roots, but they can also infect cereal roots, and that makes it tricky as well. From a breeding side, it's obviously more problematic if you are dealing with a disease complex. So you have 3, 4, 5, 6, 7 pathogens, because when you do resistance breeding, you have to do it pathogen by pathogen. So if root rot was just caused by one organism, well, you only had to go through the breeding process for that one pathogen. But because we have more than one, it's just so much more complicated. And we are mostly concerned about the 4 I've highlighted here in the red square. So in addition to Aphanomyces, the Fusarium arenaceum, Fusarium solani, and Fusarium oxysporum. And they really, so as I said, they usually co-occur in fields, or often co-occur in fields, and they have additive effects. So you see more severe disease because there is more than one organism. And to make things even a little bit more complicated, one of them, Fusarium solani, is actually a species complex. So that group of organisms that have the label Fusarium solani actually consist, and that is really fresh. The most recent research we have done here that Fusarium solani itself, again, consists of at least 4 species. And so you're not just dealing with Fusarium solani, but with these other 4 species. And if that wasn't yet complicated enough, one of them is the true Fusarium solani, and unfortunately that can also infect humans. And because that happens, it makes research much more complicated, because you will have to adhere to many more biosafety conditions when you work with this particular pathogen. So we've just sort of identified these 4 species, and so we will try now to look at whether all of them are virulent on pea and lentil, or whether sort of they are a little bit more specialized, which will again guide us in the resistance breeding. We will focus in on those species that are most virulent on pea and or on lentil.
No, yeah, obviously, I mean, this is, not simple. These pathogens, I mean, there's so many intricacies here that you have to look at. There's different species, etc. But you've got a lot of great tools in your toolbox, and one of those is something that I wanted to specifically ask you about today, and that's something called RNA inference. And this is a method that you're using in your work. And can you talk a little bit about RNA inference and then the potential it has for in-field disease management?
Sure. And what I'll do is I'll try and animate. Can I animate through your?
We should see whatever you're seeing on your screen. So yeah, if you click play, it should.
I can't animate here. So let me just check one thing. Sorry about that.
That's okay. If it doesn't animate, that's all right.
Oops. I see. Let's try this again. Sorry. Oh, here we go. Okay. Okay. So I'll try to explain RNAi. So RNA is a small molecule that all living organisms have, and it's sort of the molecule that translates the information that is present in a gene into a protein, and the proteins are then the building blocks for enzymes, for structures, for everything that really makes a living organism. So on the left-hand side here, this large yellow sun, I guess, is supposed to be my seed. And here on the right-hand side, I tried to sort of paint a spore, and with a little tube, which we refer to as the germ tube. So what usually happens, so the fungus, so the spore germinates, produces this germ tube, and then it makes contact with the seed. And if the seed has no resistant or hasn't been treated with a seed treatment, it will then be able to infect the seed. So typically, what we do is we try to protect the plant, whereas RNA interference works in a very different way. So that maybe the way we can think about it is that the interaction between the seed or the plant and the pathogen is really a little bit like warfare. So you could imagine that the pathogen has a lot of factories in which it produces weapons, and then it uses these weapons to bombard the plant or the seed in order to basically invade it and then start growing in these plants. And then often the plants have natural resistance, or the plant breeder has bred resistance into these seeds, so there are defense mechanisms, or as a grower, you put a seed treatment on or apply a fungicide. So that's sort of the traditional way of how we protect the plant. Now, RNAi is quite different. So maybe the way you can think about it is that this RNA interference, the RNA are the machines in this factory in the pathogen that produce the weapons. And as the name says, RNA interference, it interferes with the machinery in the factory, and as a result, the pathogen can actually not produce the weapons, and because it can't produce the arms, it cannot bombard the pathogen, the plant, and because it can't do that, it is unable to invade. So it's quite a different approach in that you really try to shut down the pathogen in producing weapons that enable it to infect the plant. Now I'm showing this here as a spray inoculation, but obviously if you're dealing with a root rot, it would probably not be a spray as such. It's more likely going to be a seed treatment or soil drenching or an application method like that. And sort of the formulation would be part of that research program here as well. So the neatness is that it is quite a different approach. It can be, or it is usually very specific to the pathogen, which has advantages in that it can be very targeted. But again, obviously, because we are dealing with a root rot pathogen, you will have to develop different RNAi approaches for the different pathogens. So
Yeah.
it's really unique and has great potential. So this work will be done by a colleague at Canada, Steve Robson, and I understand he has been working in the last few years on Fusarium head blight, and this year will sort of have the first field screening, so trials in the field. So it's very exciting.
It is. And, speaking of people you're working with, I mean, there's a lot of firepower behind this. I mean, you're working with a lot of different people. And before I let you go, can you just talk a little bit about, a lot of the partners you're working with and how that's really adding a lot of value to this?
Yes. So, I mean, as I said, sort of the framework that was given to us was that it was supposed to be a really comprehensive proposal, and no one research group can really tackle all areas at the same time. And so we contacted people at the National Research Council here in Saskatoon, and then also a few research centers of agriculture and agri-food Canada, all of them with slightly different expertise, and that is really what enabled us to put together this comprehensive proposal. So I think in total there are about 11 scientists here who will collaborate on this project. And I'm also listing sort of all the funding bodies. So, I mean, it's sort of very comprehensive in terms of the collaboration on the research side, but it's, I think, also very good to see that in addition to the Saskatchewan Ministry of Agriculture or the prairie grower organizations involved in pulse crops and ADA in Alberta have also decided to contribute to that, and that just allowed them to pull together that kind of funding to support this comprehensive proposal. So it's a really great initiative.
Sorry, I was on mute. So before we hear from Tyler, Sabina, once all this research is over with and finished, what's your biggest hope in terms of what you hope will come out of all this?
Now, I know there are people who, in particular on the funding side, who hope we will have solved the problem of root rots. I think that is a little bit optimistic, but I'm really hoping that at least we have identified some short-term solution, sort of on the way to developing more effective long-term solutions, but really that there is something tangible there for producers, because right now the tools they have are very, very, very limited.
Yeah, no, this will be a huge help. Thank you so much, Sabina. Hang on the line in case we get some questions at the end. Thank you for this work. Looking forward to seeing what comes out of it. And we will chat again. Thank you so much.
You're most welcome. Thank you.
Now, like I say, we are just going to hear from Tyler. Tyler's talk was pre-recorded. I chatted with him the other day. It's about 10 minutes long, so sit back and enjoy his talk about what he's working on with regards to wheat. Tyler's a real pioneer in the realm of making wheat resistant to wheat midge. And one of the ways his team is doing this is through smell, making wheat plants literally smell bad to the wheat midge, not to people, but when the insects land on the wheat, it actually smells bad to them and it deters them. Really fascinating stuff. And I'm really oversimplifying it. So Tyler's going to talk about that now. And once Tyler's done talking, we'll hear from Horatio before we wrap up for the day. So again, sit back and enjoy my chat with Tyler. How are you?
Doing all right. How you doing, Mark?
Excellent, excellent. Now we're talking today about some really cool research that you are doing that is featured in our Public Plant Science Showcase, and that's some work you're doing regarding wheat and wheat midge. Now you are an entomologist at AAFC Saskatoon, and
right.
you have essentially done some research to sort of figure out how to make, and I know I'm greatly simplifying things here, but how to make the wheat plant, smell bad to the wheat midge insect so that the insect will to be deterred from landing on the wheat, because I guess somehow to the insect, the wheat actually has a bad odor. And I know I'm really simplifying that, but can you talk a little bit about that, how this works? How can wheat smell bad to an insect?
All right. Sure. I'll walk you through this, Mark. So first of all, though, I'm going to shout out to my co-authors and the guy who's actually been doing the work, Dr. Chaminda Wiradana at the University of Manitoba. So he's Alejandro Costamagna's postdoc. He actually came out of the same PhD lab that I did. And so a chemical ecology lab, Dr. Maya Evenden's lab at the University of Alberta. And so Chaminda's been working on this thing called oviposition deterrence. So this is your making the wheat smell bad. So I'll qualify it with, we're not actually making the wheat smell bad. Some of the wheat just smells bad. Yeah. So you're a tiny little insect. Pretend you're a tiny little insect there, Mark. You're not quite as orange as a orange wheat blossom midge, but when you're a little insect and you got to find your way to a plant, vision helps, but also smells help. And so they like to smell their way to a plant. And if you're looking at an insect, you've got these tall antennae on their head, and they have little scintillae coming off the antennae. And they use those to smell volatiles coming off the plant, so volatile chemicals. So all of us, including plants, when we're respiring, our cells are making compounds and breaking down compounds, and some of those get released as volatile compounds. And so we started noticing, scientists started noticing that some wheat just got less eggs laid on it by the wheat midge. That's oviposition. I was going to say less oviposition, but I should qualify. Oviposition means egg deposition. So the female likes to lay eggs less on these plants than on other plants. So you'll have a bunch of experimental wheats in a field, and one of them, for whatever reason, just gets less wheat midge pressure for no good reason. So totally independent of the SM1 gene and its mode of action. SM1 gene is a form of antibiosis that actually goes after the larvae. And so the larvae don't like that, and they stop feeding. Whereas the oviposition deterrence is a plant defense called anti-zoonosis. So it just makes the plant sort of unpalatable for those insects. Say, let's go back to you being a wheat midge, and you've got your antennae, and you're flying around trying to find a place to put your eggs, and you start smelling this plant, and you're like, it kind of smells right. I'm picking up 3 different volatile organic chemicals that I like the smell of, and they seem to be in the right ratios. So, you know the old equalizers where you're like looking at sound and things are going up and down like this? That's what a ratio would look like when you figure out what the ratio of these volatile organic chemicals are. So you got bars like this. So if your 3 bars are in the right heights, the right amounts, then the wheat midge is like, yep, I like this wheat. It smells really good to me. Woof, in we go. I'm going to land on it. Whereas other wheat, those bars are in the wrong ratios. And it's like, wow, it doesn't really smell very good to me. So the ratio is off. And so the work that was done was actually years and years ago. And I ran into Burkett at a conference, and I said, why did you have to do the air entrainment on wheat for like a week straight? And he's like, well, wheat's not very smelly. And so for us to collect enough of those volatile compounds coming off the crop, we had to do it for a whole week straight. So Chaminda at the University of Manitoba is probably doing these air entrainments for about a week straight. And then you pull the air off of the wheat that smells good, and you pull the air off of the wheat that doesn't smell good to the wheat midge, and then you compare the amounts of those volatile compounds. And so these plants that were showing up with oviposition deterrence, they have a different ratio of these compounds. One in particular is like a typical, we call it a green leaf volatile. So if you've ever mowed your lawn, which you probably have, you know that cut grass smell? Those are green leaf volatiles being released by the plant cells being lysed by your blades. But during normal respiration, these green leaf volatiles also get released. And so that is what this wheat midge is cueing in on. She's coming in and, oh, this wheat doesn't smell very good. And often the wheat that doesn't smell very good is bad for her offspring. So female wheat midge, if she lays eggs on a post-anthesis wheat plant, that means after it's done flowering, her offspring don't have a very good chance of surviving. So their survival goes way down on post-anthesis wheat. So she tries to avoid laying eggs on post-anthesis wheat if she can help it. And she likes to go for pre-anthesis wheat. So no flowers yet. You don't see any anthers on the wheat spikes. And that is optimal timing for her to lay eggs and have her offspring survive. So what Chaminda was able to figure out was that the oviposition deterrent plants actually smell like post-anthesis wheat plants. So plants that her offspring are not going to do very well on. So like a good mom, she's like, I'm not going to feed you this terrible diet because you're probably not going to survive, and I'm going to waste all of my eggs on this wheat. So oviposition deterrence in a wheat plant tricks these female wheat midge into saying, I'm not going to lay eggs there because, yeah, my offspring are not going to survive. Even though the oviposition deterrent wheat is in its pre-anthesis stage and probably would be just fine for her offspring to develop on. But because it smells like wheat that it wouldn't be a good idea for her to lay eggs on, she doesn't. And we get reduced oviposition, which we call oviposition deterrence.
So correct me if I'm wrong, but so for, say, plant breeders, I'm thinking, and I'm a sort of a layperson.
I was going to talk about the plant breeding aspect, so go ahead.
Well, yeah, that was my next thing. So for plant breeders who say they want to make new varieties of wheat that can deter wheat midge, so I'm thinking, and correct me if I'm wrong, I'm a layperson here, but they can then take certain kinds of wheat that you've identified as not smelling good to the midge and then use those specific types of wheat maybe in the breeding of new lines so that they can create specific varieties that are maybe more likely to deter the midge. Is that kind of how this work can be used?
That's exactly how it's being used, yeah. And so we've got 2 projects on the go right now where we're trying to get that oviposition deterrence into that next generation of wheat, trying to get all these traits together. So think of defense as a pyramid. So at the top, you've got something like oviposition deterrence that just keeps the female off the plant. Then we're also trying to look for things like mechanical deterrence, things like awns and hairy glooms. So when she does get to the plant, she's like, yeah, I don't want to land on this plant because it's spiky. And then that last bottom bit of the pyramid, that's your SM1, your antibiosis. So it is a bad taste. So she lays eggs and her offspring hatch and they start tapping into the seed. It is like the last best line of defense against the wheat midge, and those larvae stop feeding, but the kernels still take a little bit of damage. So think of that as pyramiding, and this is what the breeders are trying to do, is pyramid all of this defense into one wheat plant. Now this oviposition deterrence, though, is being a little bit difficult to pass along. SM1 is that last line of defense, and it relies on just the one gene. So you can get a marker gene for that, and you can say, using this marker gene, we know that these plants are now expressing SM1. Oviposition deterrence seems to have multiple sites, and you've got wheat that has 3 different genomes crushed into one plant. It's very difficult to try to nail down where that oviposition deterrence is coming from. And it's possible that the oviposition deterrence is really hard to nail down to 1 or 2 genes because it's reliant on a number of different genes and a number of different metabolic pathways that are creating these metabolic off-gassings that we'll call a headspace around the wheat that she is smelling as she's trying to make her decisions. So far, we've found several QTLs, or quantitative trait loci, areas of the genome where oviposition deterrence seems to be happening, but they're all over the place, and there's a few of them. So then our next challenge is, okay, well, which one is actually causing the best oviposition deterrence, or do we need all of them in one plant? And yeah, so it's not as straightforward breeding-wise as SM1 was.
I can see some potential for some really neat variety names. Like you could go with a serious name like AAFC deterrence, or
Yes.
you could go the silly route and have like AAFC smelly or AAFC stinky or something like that. So there's a lot of potential for that kind of thing.
Yeah, let's avoid AAC stinky wheat though.
No, this is neat. Thank you so much, Tyler, this is really interesting, and looking forward to seeing what comes out of it. What's the timeline here? I know this is like speculating down the road, but when we might see like varieties that have this, included in them, is that something you see like in the near future potentially?
It's already out there. So
it's already out there. Oh, okay.
Yeah, some of the oviposition deterrent lines that were originally identified like years ago, those have been used as parent lines, and we've got like market ready varieties out there that likely have this oviposition deterrence. So our next steps, though, now that we know so Chaminda was basically trying to answer the mechanistic question: what causes oviposition deterrence? And so now that we know that, we can look at these other varieties and say, okay, are they presenting with those bars that look like oviposition deterrence? Do they smell like post-anthesis wheat, or do they smell like pre-anthesis wheat, which is still attractive? And so that, those are next steps, screening process for things that are already out there. But according to our breeders, they know that some of these varieties that are already being planted in the fields likely have oviposition deterrence in addition to SM1. So yeah,
nice.
These things are already happening out there.
Yeah, that's exciting to have varieties that are already out there being grown, and then now it's just a matter of saying, okay, like how well are these working and how are they working? And that's really exciting to be able to have that, to be able actually to go out in the field and actually see how sort of how the fruits of your labor are actually working. So that's really cool. No, thank you so much.
Yeah, you're welcome, Mark.
We will move over to Horatio. Welcome, Horatio. How are you today?
Yes, very good. Thank you. And thank you for having me here.
No, thank you for taking the time. And this has been something I've wanted to talk about for a while now, is soybean cyst nematode. Now, this is something you're working on at Ohio State University, and it's something we have a big interest here in Canada because SCN, as it's called, isn't really super prevalent here in Canada right now. There have been 1 or 2 instances of it, but it is moving north. And so I'm hoping you can illustrate for us, before you talk about exactly what you're working on, what soybean cyst nematode is and why exactly it's so destructive.
Yes, sure. So I had a presentation ready. I don't know if you can share that.
I can. One moment. There we go.
Excellent. Okay, perfect. So I'm putting this QR code here to, if you're interested, to send you to the SCN Coalition website. It's a website that really, aggregates a lot of information about soybean cyst nematode in general. management, what it is, life cycle, very nice infographics, videos about damage and biology of these pathogens. So a very holistic website that you will get a lot of information. So I encourage you to get your phone, snap that QR code, and explore a little bit about that website, the SEN Coalition. Next, please. So the question that Mark posed was, in a nutshell, what is soybean cyst nematode? And like I was starting to say, it's an animal. It's an animal that can clearly penetrate root system and cause yield reduction. The nematode will overwinter as an eggs, like you see there in the image. These are all pictures that we collect in our lab. The nematode then hatch from the egg, and it's the only infective stage, the only stage that the nematode will be able to infect roots. This is the second stage, juvenile. Like plant parasitic nematodes, they go through 4 different stages before they become adult. You can see that we stain roots and stain the nematode as well, and we can see that the nematode penetrate roots and migrate to the vascular cylinder, where it will start releasing some effectors. I mean, this is an animal, again, an animal that is releasing effectors into plants that will modify cells and take control of the plant machinery. It will start modifying cells to then feed the nematode that now becomes sedentary. It cannot move anymore. And it will continue developing, going through all the different stages until they reach adulthood. The males will regain the vermiform condition. As you can see, there's a long males that look like a worm getting out of the root and gonna go and localize females and fertilize females. And the females will lay eggs on that gelatinous matrix that you can see on the rear of those lemon-shaped females. On average, 250 eggs. They will eventually fill up with eggs and die. The dead female is called cyst. These dead females will oxidize, change color into a darker one, become very resistant, and protect all those eggs within. And this is a way that the nematode will overwinter and will survive in the soil for long periods of time. Next, please. How do we know we have it? Well, when there are abiotic conditions that can interact with the nematode, we can see stuntingness of the plants. We can see areas where the plants die prematurely, yellowing, but really happens when we have other factors like hydric stress, very sandy soils, nutrient deficiency, another pathogen that may interact. But the root will not lie. The root is where the sign is where the nematode will be found. We can pull those roots very gently and look with naked eyes, those lemon-shaped, yellowish in color, whitish sometimes, females attached to the root. Very, very tiny compared to a nodule. Nodules are larger and bigger than we want a root to have a lot of nodules. Those are the nitrogen-fixing bacteria interaction with a legume. Next, please. But the situation is that this nematode can infect the plant, modify those roots, and produce yield reduction between 15, 50%, even more. And in the Midwest and in areas where we do plant a lot of soybean and these pathogens have adapted to the crop, if you can hit one more, this is really the nightmare that we have to deal with. There's really no visible symptoms in many cases. So we can compare a susceptible and a resistant cultivar, as I'm doing in the right figure here. Absolutely no visible symptoms, yet when you go with a combine and harvest that, you can detect over 30% yield reduction. And this is, in my opinion, why these pathogens really remains, that's fine. You read my mind there. You need to go to the next one. Remains the most destructive pathogen. No matter how we quantify this, and we do it, we get together with plant pathologists and we put out reports, and these reports have been coming out for decades now. And if you go to the next animation here, the soybean cyst nematode just ranks the #1 yield robbing pathogen. And if you grab the next 5 pathogens and calculate how much they're reducing yield, it's really the soybean cyst nematode that takes the lead. You can just go back and stay on the previous one. So, in 6 minutes, that's the short answer for what's the soybean cyst nematode and why it's so destructive.
And now this is something, Horatio, that is obviously been present in the United States, but is now moving up our way into Canada, correct?
Correct. Yeah, so if you go to the next slide, I have a it's already one of the, classic literature in terms of distribution of soybean cyst nematode. And if you visit this article, and there's a new one coming hopefully soon, updated, but this is the most recent one that we have. It shows the distribution of soybean cyst nematode in United States and in Canada, right? If you hit the next the next animation here, you will see I purposely brought one of the tables from this article that specifically show areas where in Canada the nematode has been detected. But it's very interesting here. If you look at this map, and if I were to tell you that this map is really a map showing the distribution of where soybean is grown, you will not doubt that, right? This is exactly the pattern of soybean production in North America. So it's very clear that the nematode is following the crop, and wherever a new area opens up for soybean production, it's going to be a matter of time when soybean cyst nematode will be detected. Above damage thresholds will be quantified, and yield reduction will take place.
Now, Horatio, there's currently a lot of things that are being done to help kind of battle soybean cyst nematode. Can you just walk us through quickly a few of the current practices that are being used?
Yes, sure. And again, that's a interesting question, right? Because I'm telling everyone that soybean cyst nematode will cause tremendous yield reduction with absolutely no visible symptoms. So, what am I really managing? And if my beans are beautiful, why should I even bother in questioning if something is wrong, right? I don't think we want to do that, but that's exactly where management should start. And the SCN Coalition put this way of raising awareness by saying a proactive management of soybean cyst nematode starts with knowing you have it. So know your numbers. That's a slogan that is associated with this campaign of raising awareness. So soybeans look beautiful, but do you know if you have soybean cyst nematode? Pull a plant, pull those roots and check if there's a female attached to the root, or more easily, in my opinion, collect a soil sample. If you go to the next slide, this is a very, common practice that we all do. We take soil sample. When we do agriculture, we will collect a soil sample, a composite soil sample, send it to the soil lab for nutrient evaluation, right? And the same way is done for soybean cyst nematode testing. So if you are already collecting a sample for soil test, take a subsample of that and send it to the nematology lab to answer the question: do you have soybean cyst nematode? Is that enough? Not really. How much you have? Because the levels of soybean cyst nematode will, help you strategize the management you will apply. If you go to the next one, please. And the reason we want to know these numbers is because, I mean, this animal can just reproduce so efficiently. Even with most of the population in one season dying, the nematode will be able to go from very low levels of soybean cyst nematode, levels that we will consider low, that will not cause yield reduction, like 200x per 100 cc of soil, cubic centimeters of soil, in one season exploding to, you know, above 10,000, which will cause yield reduction in a susceptible soybean. Next, please. And this is another tool that the SCN Coalition has recently launched, the SCN Profit Checker. Once we know those numbers, what can we do? And another tool that helps raise awareness, start conversations with growers, is this one here. You can input the numbers of soybean cyst nematode that the lab, return to you, a reproduction on one of the sources of resistance, PI 88, percentage of sun and pH, and it will give you an estimate of how much you're losing due to soybean cyst nematode. And again, this can raise awareness, make you realize that, okay, my beans are looking beautiful, but I may be losing a lot without knowing, right? And that will, encourage you to take some of these different management strategies that we will talk just right now in the next slide. So very briefly, and trying to wrap up here in this short time, an integrated management approach is what we recommend. And this is not news, right? When we talk about different pathogens, we normally encourage an integrated management approach. Active management, know your numbers, know your types. Not all SCN populations behave the same way. Some will be able to reproduce on specific sources of resistance. So we should identify and know our population type. And this is something that a nematology lab can do. Next, please. The triple R, which is one of the easiest way to remember the best management strategy for soybeans, is nematode. Rotate, rotate, and rotate. I mean, very easy to remember. Rotate to a non-host. Planting corn, small grain, sunflower, cover crop, anything that the nematode cannot reproduce following soybean will decrease populations of nematodes in the field. Rotate resistant varieties. We don't want to train the nematode to feed on the same genetic material. Most of the 90% of the commercially available soybean that says resistant to soybean cyst nematode have these PI 88 source of resistance. The third rotate is try to find different sources of resistance. And then last, if you hit again one more time, consider a seed protectant. Most of the time, these nematode protectant seed treatment, if maybe not so effective in reducing population of nematode, will enhance the overall soybean root health. There are other organisms out there that may interact with the nematode and cause more disease. And we want to protect holistically the root system of the nematode. And with that, if you just go one more and maybe two more, this is just reiterating things, infographics that you can find on the SCN Coalition website. But here, we present a 6-year rotation program that can be used to manage soybean cyst nematode. Make it look great, as you can see on that left image, but keeping those yields up.
Thank you so much, Horatio. And actually, now that we're done that part of your presentation, before we wrap up for the day, I actually wanted to talk with you about some really cool work you're doing. You just took part in a project to develop, I believe it's the first trait in, I believe, 6 decades that is the first SCN resistant trait that has been developed. And you actually took part, I understand, in the development of this. Can you tell us a little bit about that? Because like Sabina was talking about developing these new tools in the toolbox, these new weapons in the war against these pathogens. This sounds pretty significant, technologies like this.
Yes. So my research program was not part of developing that. My contribution was that I was present when this information was released to the media. So I did a lot of the, biology of the soybeans is nematode, and kind of, bringing the whole stage to why this pathogen is so important and so destructive. Hopefully the same way that I just did. And then just presenting the limitations that we're running into. So I do work a lot with growers, and even though we're saying use resistant, seed protectant, we are still noticing that populations of the nematodes are, adapting and developing on our best sources of resistance. Seed protectants are not doing, the work that we expect. So, I was very happy to be part of that interaction, but really, I think the trait that you're talking here was developed by BASF. This is a company, well known. And the interesting thing here is that this is a transgenic trait. It's the first transgenic trait that we will now be able to use to evaluate a different way of managing soybeans as nematode, because this trait is a cryoprotein like the BT. So probably you are very familiarized with BT. This is a type of cryoprotein that the nematode, by ingesting it, this protein will interfere in its digestive system and kill the nematode. So the beautiful thing here is that this trait can be put into different backgrounds. So we have our sources of resistance that now we're using and trying to optimize, because we know that the nematode is adapting and reproducing. Well, now we can add this trait to these available and commercially available soybeans that we have, right? Sometimes we like particular traits in the sense of herbicide and weed management. So, we want soybean seeds with particular traits that allow us to spray herbicide. Well, now we can put this nematode trait on our favorite soybean, right? So this is very exciting. There's already trials going out. There's already data being generated. And they are expecting, pending on regulations, to release this to the public in 2028. So very exciting news from the private sector. Other researchers, just to add a little bit on other stuff that is going on in terms of future management to soybeans as nematode, are discovering how these different genes work, the mechanisms actually of resistance. So we hope in the future that we will move away from sources of resistance and talk more about combining different genes or lack of genes. So with gene editing, for example, we could silence some of these genes that are important for pathogenicity or for the nematode to actually infect. So these are great discoveries that are happening, and you will find all these in the SCN Coalition website. So in my lab, we do explore a lot of these in the lab, both in molecular in the greenhouse part of the lab, but also take those results to the field. Ultimately, we wanna see what we are doing in terms of improving yield, improving production, and reducing nematode population in the field.
Yeah, it's definitely exciting, and for you to have tools like this that you can use in your work to help create these new milestones in the battle against this, that's always coming in handy, I'm sure. I'm just going to bring Sabina back onto the stage here quick before we wrap up for the day. And Sabina, I'm sure for you, when you think of what's coming down the pipe in the future when it comes to root rot in pea and lentil, and can you give us an idea of, in 5 years, 10 years? I mean, I know we'll never have that magic variety that's resistant to everything and that solves all the problems. That doesn't happen in plant breeding. But Sabina, what for you is, that ultimate variety that you think you could, through this work, potentially bring to the market one day that growers could actually have and use?
I mean, we have in pea, we are sort of furthest ahead because we were fortunate in that people elsewhere in the US and also in France had already done a lot of the legwork. So we are actually very close to having germplasm that has Aphanomyces root rot resistance, but obviously to really have an impact in the field, we now have to combine it with Fusarium, right? So I do hope that within the next, let's say 5 years, we will have lines that have combined resistance, but obviously it then still takes a while for these lines to go through the registration process, and then you have to do seed increase until they really become available to growers. With lentil, we are further behind. We have to do more of the legwork. But we have identified sources of resistance, and Horatio was mentioning those as well. So that is really important. And then longer term, I think, as with every host pathogen system, the more we learn about the biology and the molecular side of it, the easier it will be to use these modern technologies to make breeding faster and more efficient. And I think that will happen with the root rots as well.
Yeah, I'll give you the last word, Horatio, before we sign off here for the day. And what are you most excited about when you think about, the future, what the future holds in battling something like SCN?
It's exciting that we are having new tools coming, that we have data that is showing very promising outcomes, in the sense that, ultimately our mission is to help the growers and to produce sustainably more food, right? But when I say, help the grower, it sounds like a one-way street, and that's absolutely not the case. The work that we do when we interact with grower, for me, is so valuable. Most of the very significant questions that my program is working and developing, they all stem from conversations with grower, visiting the farms, visiting the producers and stakeholders, right? So I'm mentioning growers, that for me is my ultimate stakeholder, ally, collaborator. But in that process, industry is so important too. And if we are all on the same page, and if we are all opening these two ways of communication, it's the interaction and network is great. And I think that's how we can move faster to that ultimate goal of safety and food production for the world.
Well, before we wrap up, that's a good point. This is the retail strategy webinar, so we try to give advice for retailers, people who sell seed, and so I imagine, like you say, that feedback works both ways. You need feedback from growers, and so, Sabina, is there a way that retailers, people who sell seed, who work with growers, on the ground, that they can help maybe solicit some of that feedback and communicate that back to you? Do retailers have a role to play in this?
Oh, certainly, certainly. And I think if you look specifically at the pulse industry, I think they have developed a fairly good network where there is sort of a stream of information going both ways along the whole value chain, right? And so it's not just the producers, but also sort of the seed commercializing organizations that get together and then develop policies really and strategies and priorities, what research is really needed to ensure that the pulse crops remain competitive in the international market. So I think there is that infrastructure out there and everybody's involved.
And yeah, Horatio, what's the easiest way that, say, retailers, that they can get, information to you that maybe they get from growers, and if they can communicate that right to somebody like you who works in the scientific department, I would think that would be really useful. And what's the best way they can do that?
That's a good question, right? Because the easiest way will be, email me and I'll be able to reply, right? But these sites such as the SEN Coalition, for example, it's a great example of, private-public partnership. You go there, you'll find a list of the different universities with their specialists, plant pathologists or nematologists that you will be able to contact. But then also all the other private companies and collaborators that also through that network hub, you will be able to connect. And I think that's one of the missions and visions of the SEN Coalition, trying to be that catalyzer of place where you can go and get a lot of information, but also identify, the key players that you could reach out to and engage and start a process of conversation and discussion and why not collaboration, right?
Exactly.
Could I maybe add? Yeah, I just wanted to add something. Now, obviously, I'm working here at the Crop Development Center, so we are developing varieties, and sort of as a someone in the business of commercializing seed, we don't commercialize our varieties ourselves; they're tendered out, so they will directly interact with the CDC, and that's sort of where often the feedback comes. They will pick up varieties that they feel have add value to producers, right? And so that's at the lowest level where this interaction happens.
Yeah, that's a good point. No, thank you, Sabina and Horatio, for your time today. This was a great discussion. And for those who are hoping to see my chat with Tyler, don't despair. The recorded version will include my talk with Tyler. So that's a good incentive when you get the email about saying that the recorded version is now online. You can watch that. You can fast forward if you want right to my talk with Tyler and watch that in its entirety. So that will be coming very soon. Horatio and Sabina, thank you so much for your time today. Again, I just want to thank our sponsor, CCAN, one more time for their support. And I'll thank Tyler as well for having his chat with me the other day. Again, that will be in the recorded version. As I said, this webinar was recorded and will be on our website, seedworld.com, in the coming hours. Again, I just want to thank our audience for spending your lunch hour with us. This was a great chat. Again, Horatio, Sabina, thank you very much. Have a great day.
Thank you.
Thank you.